This method can help answer key questions in the stem cell engineering field, such as how cellular microenvironment alter the cellular phenotypes and function. The main advantage of this technique is that it provides the multiple artificial regulatory environment for the cells in a single plate, which we cannot perform with a conventional cell culture microtiter plates. Demonstrating this procedure will be Koki Yoshimoto and Risako Sakai, technicians from my laboratory.
After creating 3D images of masks for nanofiber arrays and molds for microfluidic structures according to the text protocol, use a 3D printer to print the masks and mold. To prepare polymer solutions for electrospinning, dilute 13% PMGI liquid with tetrahydrofuran to 9%. Dissolve 0.08 grams of PS in a 1:1 volume ratio of THF to dimethylformamide, and use the liquid reagent to bring the volume up to the final 1 milliliter of 8% weight by volume PS solution. Dissolve 0.1 gram of GT in water, acetic acid, and ethyl acetate, and use the mixed solvent solution to bring the volume up to the final 1 milliliter of 10% weight by volume GT solution. Next, using a magnetron sputtering machine, deposit a 5 nanometer thick platinum layer on a polystyrene base plate, which serves as a cathode in the electrospinning setup. Load each polymer solution into a 5 milliliter syringe equipped with a 23 gauge stainless steel blunt needle. Then, anchor the syringes with the needles on a syringe pump, 12 centimeters apart from the collector of the electrospinning device. Connect the syringe needle